Hi everyone, in this video we are going to see about what is Genomic Library. Genomic Library is nothing but is a collection of overlapping DNA fragments that together make up the total genomic DNA of single organisms. The Genomic Library represents an entire genome of an individual animal, plant, bacteria or virus. It is a collection of cloned segments of the DNA containing at least one copy of every gene from a particular organism. The DNA is stored in the population of identical vectors, each containing a different insert of DNA. In order to construct a genomic library, the organism's DNA is extracted from the cells and then digested with the restriction enzymes to cut the DNA into the fragment of a specific size. The fragments are then inserted into the vector using DNA ligase. Next, the vector DNA can be taken up by the host organisms, commonly a population of Escherichia coli or yeast, with each cells containing only one vector molecule. Using a host cell to carry the vector allows for easy amplification and retrieval of specific clones from the library for analysis. There are several kinds of vectors available with various insert capacities. Generally, libraries made from organisms with larger genomes require vectors. Featuring larger inserts, thereby fewer vector molecules are needed to make the library. Researchers can choose a vector also considering the ideal insert size to find the desired number of clones necessary for full genome coverage. Genomic library are commonly used for sequencing application. They have played an important role in the whole genome sequencing of several organisms, including the human genome and several model organisms. The common vectors used in the genomic library constructions are plasmid, it is up to 10 kb, insertion of size up to 10 kb, modified lambda phage up to 20 kb, insert or 40 kb for cosmids, further modification of phage. Then the artificial cloning vectors such as BAC, bacterial artificial chromosome vectors, up to 100 to 150 kb inserts. Then yeast artificial chromosome up to 500 kbb inserts. Then let's see about construction of genomic library. Construction of genomic library involves creating many recombinant DNA molecules and organisms. Genomic DNA is extracted and then digested with the restriction enzyme. For example, with very small genomes approximately up to the size of 10 kb, the digested fragments can be separated from the gel electrophoresis. The separated fragments can then be excised and cloned into the vector separately. However, when a large genome is digested with the restriction enzyme, there are far too many fragments to excise individually. The entire set of fragments must be cloned together with the vector, and the separation of clones can occur after. In either case, the fragments are ligated into the vector that has been digested with the same restriction enzyme. The vector containing the inserted fragments of genomic DNA can then be introduced into the host organisms. For example, in the species of E. coli, it has its own DNA or genetic component. When we take the genetic component and degrade it into the smaller fragment by the restriction enzymes, endonuclease, and take each of the genetic component and clone it with a different vector and put those vector back into the host cell, grow those cells in the Petri plate. Now the Petri plate contains different types of colonies. Differential segments of E. coli or other cells is formed. This particular Petri plate is act as a genomic library for E. coli. Each of the colony consists of a particular set of genome. From the chromosomal DNA by the restriction endonuclease, formation of gene fragments, and insertion by the insertion of vector, there is a formation of recombinant DNA in the E. coli. The steps for construction of genomic library are extract and purify the DNA, digest the DNA with the restriction enzyme. This creates a fragments that are similar in the size and each containing one or more genes. Insert the fragments of DNA into the vector that were cut with the same restriction enzyme. Use the enzyme DNA ligase to seal the DNA fragments into the vector. This creates a large pool of recombinant molecules. These recombinant molecules are taken up by the host bacterium by the transformation, creating a DNA library. This is the image of construction of genomic library. Uh, in this picture, there's the human DNA, extraction of human DNA that cleave with the restriction endonuclease. The fragments are formed. There is the genomic DNA fragments, and the DNA fragments are inserted into the suitable vector by using ligase. Then by using ligase there is a formation of recombinant DNA molecules. Then the plasmid into the bacteria. This are known as genomic library. Thank you.